Right. Good afternoon, and thank you very much for coming. And welcome to the Nai-Wen Chi Memorial Lecture. And thank you to Dr. Altshuler for being the speaker and the donors who made the lecture possible. I will just give very few brief remarks about Nai-Wen Chi. Nai-Wen died on August 10, 2021, after a brave 3-year struggle with metastatic cancer. He is missed by many of us, especially his family, his widow Lily, and their sons Dylan and Tyler. Nai-Wen was born and raised in Taiwan and graduated summa cum laude from the National Taiwan University Medical School in 1986. After serving 2 years in the Taiwanese army, he married his medical school classmate Lily and immigrated to the US to earn a PhD in biochemistry in 1994 from the Harvard Medical School. Nai-Wen then did his residency and endocrine fellowship at the Massachusetts General Hospital in Boston. And in the year 2000, he joined the UCSD medicine faculty in the Division of Endocrinology and Metabolism. Nai-Wen was an excellent physician. We attended together the VA endocrine clinic for several years, and I was amazed about his comprehensive medical knowledge. His scientific effort focused on how insulin stimulates the uptake of glucose into heart and fat cells, and he explored the not well-studied role of enzymes called tankerases, which are multifunctional poly-ADP ribose polymerases. Nai-Wen was greatly appreciated as a friend and colleague, teacher and scientist, and he was also an accomplished classical pianist. I mean, he played for us at some division meetings. For many of us, his most outstanding feature was his generous and giving nature. Thank you for attending.
I'm Amit Majithia, faculty in the endocrine division. And I wanted to say, when I was being recruited to UCSD, Nai-Wen Chi was co-chief. And so I had a chance he was the first person I interacted with and presented UCSD. And, you know, as you've heard and you will hear, that he was incredibly open and kind and generous. And when I came here, he didn't forget about me. He went so far as to give me lab equipment from his own laboratory to get my lab set up, which we still use today. So I'm very grateful to him for being here. Now it's my great pleasure to introduce Dr. David Altshuler, human geneticist, endocrinologist, and currently the Executive Global Vice President at Vertex Pharmaceuticals and Chief Scientific Officer. Prior to his role at Vertex, Dr. Altshuler was for many years a professor of genetics at Harvard Medical School, as well as a physician at Mass General Hospital. And his lab did some of the foundational work in cataloging human genetic variation with the HapMap Consortium and the 1000 Genome Projects, foundational resources that we still use today and are the basis of modern human genetic analysis. Dr. Altshuler also worked on the genetics of type 2 diabetes, which is when I joined his lab, and that gave me my start. So without further ado, please welcome David Altshuler.
It's really a pleasure to be here today. I actually remember some 15 or so years ago, I was here and visited Nai-Wen, and I gave a talk in this lecture hall, I believe. I'm going to start with Nai-Wen because Nai-Wen and I trained together. This is a picture here on the steps of the Bullfinch building at Mass General Hospital, 1997 in the spring, so 26 years ago. There are 3 endocrine fellows a year at that time. The 3 endocrine fellows, as you can see, are Nai-Wen, the tip of the pyramid, the crowning top of the pyramid. You could also see that he and I and Evan Rosen, who's now the chief of endocrine at the Beth Israel Deaconess and a professor at Harvard Medical School, were kind of iconoclastic anti-authoritarians. Pictures had been taken on the steps of the Bullfinch building by every fellowship group at Mass General for like 80 years, and we're always in your white coats sitting up formal, and we decided that we would be a little more hip and fun. But at least that's what we thought we were doing. But I do want to say, Nai-Wen and I we did our PhDs together. We did our internship together, our residency together, our clinical fellowship together, and we're postdocs, although in different labs at the Whitehead together from about 1988 to 2000. Especially as an intern, resident, and fellow at Mass General, we spent many, many hours together. And I would just agree with three things. First of all, he was in ascending order of importance. He had an unbelievable knowledge of medicine, much more than almost anyone else I've ever met. He knew everything, and he knew when it was important. You know, it wasn't just like he had endless facts. He was a very dedicated and caring physician and scientist. All that is true, but he was a generous and kind person, and he was just a terrific person, and I miss him. And I'm really honored to be here today because he was a special person for a period in my life. With that, I will move forward. And I'm going to start this talk, the beginning of the talk, and for any fellows or students, residents in the audience, a little bit of my own story, just to give you a sense of how I ended up doing what I'm doing. And then I'll tell you three stories, brief vignettes about medicines for cystic fibrosis, sickle cell disease, and something called APOL1 mediated kidney disease that we've developed at Vertex. But I start this talk on the first day of my medical school, starting medical school in August of 1986. We were handed this very famous, at least in Boston, pamphlet by a guy named Francis Weld Peabody from 1927, 95 years ago, and it's worth reading if you haven't read it. It concludes with the statement, one of the essential qualities of the clinician is interest in humanity, for the secret of the care of the patient is in caring for the patient. Now, when I first read this, I sort of thought to myself, like, why else would you go into medicine if you weren't interested in people? But I came to realize very quickly that actually there weren't actually that many people outside of perhaps the clinics who were actually interested in human beings. And if you think that I'm exaggerating that, I will show you an article that was published a month before that day. I will note that I didn't read the article I'm about to show you for 20 years, and this was written in 1927, but I create a little drama by saying that, in fact, it's the same month. This article was published in Nature in 1986 on the occasion of the now legendary Cold Spring Harbor meeting on the biology of Homo sapiens that is famous for two main reasons. The first is that at that meeting, Lou Kunkel and Stu Orkin, Boston Children's Hospital, reported the positional cloning of the first two genes ever cloned by positional cloning in a human being, which was Duchenne muscular dystrophy and chronic granulomatous disease. This was following the work of obviously Welcome Bender, who cloned the first gene by positional cloning in fruit flies, ultrabithorax, in 1983. The other thing that happened at this meeting was it was the first time anyone discussed sequencing the human genome. And so this News and Views was written in response to those advances. And everything you need to know about this article and the world in which I entered medicine and medical science was this statement: there is no scientific reason for studying man. If you actually read this article, I'm not misleading you as to what the article says. It says human beings have never taught us anything about science except for Joe Goldstein and Brown discovering the LDL receptor. Everything you can learn in fruit flies, mice, and worms. And the only thing people can do for us is there are rare mutations that the mice might eat, but in people, we might be able to find them. And that's really the only use of human beings. The article goes on to say that if the skill and ingenuity of modern biology are stretched to interpret sequences of known importance, such as that for Duchenne muscular dystrophy and chronic granulomatous disease, which I editorialized had been known for all of 3 weeks at this period of time, what possible use could be made of more sequences? And finally, those who are, it was known at that time that 99% of the genome was non-coding. And it says here, those who argue there may be meaning in the nonsense and function in the repeats are not likely to figure it out by sequencing it all. Well, I assume everyone in the room knows that the vast majority of functional sequence in any genome is non-coding. About 80% of the human functional sequence is non-coding, and it was figured out exactly by sequencing it, doing comparative genomic analysis, and then doing functional studies. I say this not to gloat, because I'm sure that I am as misguided today in terms of what will happen in 30 years as this person was 30 years ago, but to say that there really was an attitude bred on the remarkable success of molecular biology, showing us that actually worms and fruit flies and cells and bacteria all had the same genetic code, had the same protein machinery, the same histones, the same basic glycolytic pathways, that you could learn everything you needed to know from non-humans. But, the question is that really true? And does it lead to medical advances? So, I ended up in 1997 going to work with Eric Lander after I finished my medical training, and Bruce Hamilton, who's sitting right there, was the senior postdoc in the lab who showed me the way. And that's a very interesting walk you have here, showing me the way in Eric's lab. And, it's fair to say that over a period of time that I was just lucky enough to sort of be in the right place at the right time, the Genome Project advanced and we now had access to all the sequence of the human being and other species. We had that ability to study genetic variation and all that. And that certainly, one thing it has done is, instead of the focus that I grew up in, and many of you grew up in, which was there were like 100 genes in the world that explained everything. Anything you needed to know could be, if you want to know gene regulation, you look to bacteria, Jacob and Monod, that was pretty much it. If you wanted to understand how a protein worked, you looked at the proteins that have been cloned, and just saying there's a big genome out there. There's lots of stuff we don't know, and begin trying to figure out what is the function. And I think that, the acceleration of understanding human disease by genetics started with David Botstein and his colleagues publishing in 1980 the idea of using polymorphisms to be a genetic map in humans, the first genetic map in 1986, the first gene cloning in 86. And then with the advancing tools of human genetics, within 10 years, having hundreds of genes. And today, I would venture to say that if there's any Mendelian disease that's more than a handful of people on Earth that has been identified, we know the genetic basis. And I would argue, if you sequence it all and you haven't figured it out, maybe it's not a Mendelian disease. There's actually a belief system that any severe phenotype must be a Mendelian disease. That's, of course, false. But then the ability to understand the genetics of common complex traits eluded us. And I mean, if you go back 120 years, there was a debate between the biometrics and the Mendelians about how much of trait variation was Mendelian versus complex, but there was literally no ability to identify any genes for complex traits until, and linkage analysis was done and candidate gene studies, and they taught us zero. It was thousands of papers, but none of it was actually correct. It was all irreproducible because people had low standards. But when we actually started being able to do genome-wide association studies with appropriate statistics and power, and then later association studies using sequencing instead of only common variants, we were able to reveal the genetic basis, at least that which can be understood today, of common diseases. We also learned that the penetrance of Mendelian mutations was wildly overestimated by the Mendelian geneticists because of ascertainment bias. They would say, for the sake of their studies, let's only look at the most severe people. They would then find mutations, and then they'd compare them to general population and say, this is a penetrant mutation. In my lab, we did a whole bunch of studies, and others have done them now, where we actually looked at, in unselected populations, found so-called Mendelian mutations, and in many cases, their penetrance was overestimated 10 or 50-fold by that very understandable ascertainment bias, and we could recreate their results by only looking at the people at the extreme tail. So we've actually learned that the apparent distinction between Mendelian disease and complex disease was in part one group of people looking under the light at the most severe phenotypes, one group of people looking at the general, and then looking at each other. It's like the elephant, but in fact, it's a continuum of trait variation, effect size, and complexity. That's at least my point of view. So why is human genetics relevant? This is a slide from the class that Joel Hirschhorn and I taught at Harvard Medical School for 15 years of all incoming Harvard medical students, where we tried to explain to them how we thought about genetics. And there were sort of three ideas. Human genetics is relevant to medicine for three main ideas. One is you can identify or test hypotheses about human biology. Testing hypotheses about human biology is actually difficult because you can't do experiments unless you do like clinical trials. And so the ability of human genetics, meiotic randomization, recombination, having unlinked alleles allows you to get at causality and also identify through genetic mapping. The second is diagnosis and prediction. And as I've said, I think that a lot of that is misunderstood by people who think that everything is fully penetrant. But the reality is this can be useful. In many cases, it can actually be more useful than traditional measures. But it's not fully penetrant, it's not deterministic, and society is still working out how to do this. And the third is to identify potential drug targets or to inform therapeutic development. And when I think back over the 37 years now since I entered medical school, it was really the third that always motivated me. The reason I went into science in a vague, sort of poorly understood way was the idea that that Sidney Farber and people had figured out that you had acute lymphoblastic leukemia and you could develop a therapy and then you'd help patients. And they had this belief that you would be able to figure out diseases and then maybe make therapies, but it was a very vague belief. But by about 2010, as after really exciting decade or more in human genetics, the sort of emphasis of my thinking was shifting from, if only we could find some genetic basis of diseases, maybe we'd be able to do some of these things, to we found a lot of genetic clues about disease. What's the impact? And I think that there's decades to go in understanding what the hypotheses can teach us and how to understand the biology, or conversely, coming from a model system to say, can I connect the discovery I made to human phenotype? I think diagnosis and prediction is another discussion, but I became more and more focused on this question of, could we do something relevant to therapeutics using this human biology? And the more I thought about it and engaged with different people who were working in therapeutics, the more I became engaged with a different problem, not the problem of human genetics, the problem of the low productivity and attrition in the biopharmaceutical industry. So if you look at this plot, it's sometimes referred to as Eroom's law. Moore's law is the something that, it's changed the world, which is every 18 months, the number of transistors that can fit on a chip doubles. And that leads to, this thing, which is the most powerful computer. If you go back, 20 years, it would be like, the most powerful computer on earth. But Eroom's law is the opposite. It's that as time goes on, the cost and effort required to make a new medicine only increases with time. Now, if we were doing a better job, and when I say we, I mean all of us, basic biomedical science, clinical science, biotech, pharma, everybody. If we were doing a better job, this would be at least flat or maybe getting better. Because we love to talk about the awesome power of modern biology and how much we're learning, but why are we so bad at figuring out what to do? And my colleague, Mark Bunnage, who wrote a paper 10 years or so, 12 years ago when he was at another company, and I recruited him to Vertex in 2016. He's our head of research now, and he's phenomenal. He wrote this article from his other company about getting pharmaceutical research back on track, back on target. And it's just one of many articles that shows that today in the modern world, if you of all compounds, if you track all compounds that enter clinical trials, there's an IND, which if you are sort of doing basic science would be the culmination of what you're doing. I'm actually going to test something in people. The current success rate of new INDs is about 5%. So 19 out of 20 will never become a drug, okay? And if you ask why they don't become a drug, there's first what's phase 1 studies, which are safety and PK. And about half of drugs don't make it through there, but that's actually a very fast death. It takes for the drug, not the people. For the drug, it's 1 year usually, and whether or not it has safety. You do it very carefully. You start low and you work your way up. And so if you find any safety or tolerability issues, you stop. And that's sort of like a relatively rapid learning. But the next cut, which is the most severe cut, is at phase 2, what's called proof of concept, which is when you test your hypothesis. So you started 15 or 20 years ago, often as a field, saying, oh, I have a target. It's interesting. If we could only test in people, whether or not in disease X it's useful, and then you get there, it takes like 10 years to discover the drug. And then it takes another 10 years to actually go from the drug, often in some people's hands, to get to POC. And then you fail 75% of the time. The overwhelming majority of those failures are for lack of efficacy. The hypothesis was wrong. Think back to who came up with the hypothesis. It was all of us. We did the science. We said, I have a new discovery. I think this could be a therapeutic. Let's go start a company or convince somebody to work on it. 75% of the time it fails. Once you actually get to proof of concept, the likelihood of launching a drug is quite high, okay? So why is it that we are so bad at this? I believe it's because if the target is wrong, nothing else matters. Now, it doesn't mean if the target's right, nothing else matters, right? Because if the target's right, you still have to make a good medicine. You still have to test it. You have to do everything right. But think about it. If you knew in advance that this was not relevant to the disease, you're going to spend 10 or 15 years getting nowhere. So the question is, can you know in advance, or at least prioritize in advance, which targets might work? Now, if you come at this from the point of view of a disease, I'm committed to disease X, there simply may not be any targets today that have the kind of human biology and proof of mechanism that would allow you to move forward. And that's a real limitation. I think we need to do more science to figure out what those answers are. But conversely, if you look at it and say, in order to be successful in making medicines that matter to society, what I should do is find the best opportunities where we can do that. That opens up the lens, but it makes you focus in a very different way. So finally, to sort of end this part about my own sort of intellectual journey, in 2012 or so, Robert Plenge who was a postdoc in my lab and then a faculty member at Harvard, Ed Skolnick, who was at the Broad, but who was the legendary leader of Merck Research. He oversaw the development of 26 FDA-approved compounds in 18 years, which is like being the New York Yankees and winning 26. You want to know how to contextualize that? It's like the New York Yankees winning 26 World Series. And then myself, we wrote this paper, and within 2 years, Robert had moved to industry and I had moved to Vertex, and Robert's now the head of research at BMS. And our idea was simply, and actually was based to some extent on being an endocrinologist. It was like based on the idea that in endocrinology, 40% of all medicines are either hormones or hormone receptors. And why is that? It's actually because endocrinology allows you to like do experiments. You sort of had like Cushing's disease, too much steroids, what happens? You have like, Addison's disease, too little cortisol, what happens? Then people synthesize cortisol. It wasn't surprising it worked because you already knew what happened in people. So the idea was human genetics could provide you in certain cases an allelic series where you could see the difference of upregulating and downregulating the gene in people, and those would be good. And you could also see if there's any side effects or any other possibilities, and you could try and use that to select things that have a higher rate of success. So I moved to Vertex 8 years ago. And the other thing that we were really struck by was that there are new modalities. So if you're stuck with one modality, if you are what's called a platform company, so we're whatever, we do this platform, then you have to look, you're a hammer looking for nails. And you have to say, what disease can my platform do? But what we think is you actually want to merge two things. What is the human biology of conditions with severe impact and unmet need? And what are the new tools and technologies? And then are there any ways of lining them up? Those should be cases where you might succeed. And so we developed in 2015, when I joined Vertex, a very simple strategy for R&D, which was that we will only work on programs where we are certain about the underlying causal human biology and where our drug directly addresses the underlying human biology. No upstream, no downstream, no sideways, no key opinion leaders, no reviews in Nature, no reviews in Cell. It's got to be that you can actually see without any reasonable doubt that like CFTR mutations cause CF, or loss of islets due to autoimmunity is the cause of type 1, and cadaveric islets cure type 1. And that we would then try and combine that with new therapeutic modalities, whether or not we had ever worked with them before, to try and do really transformative things for patients. And so this is different than almost any other strategy in the industry because it is not a platform strategy. So most companies are like, we're a CRISPR company, we're an RNAi company, we're a small molecule company. We started as a small molecule company. We now have 6 modalities in clinical trials. We're not a therapeutic area-based company. So a lot of companies will say, we're a brain company, we're a lung company, we're a kidney company. That's a commercial strategy. The reason companies do that is because they have salespeople who interact with those doctors, not because like one brain disease is really anything like another brain disease. And then the third is what's called a Me Too strategy. That's just, I'm going to wait for someone to do something and copy them. We don't think that's such a great strategy either. So I go through this just because actually I don't think there's another company in the world that's doing what we're doing. And so it's puzzling to people. They go, I don't get it. Why are you doing the combination of cystic fibrosis, a CRISPR for sickle cell, a small molecule for pain, a cell therapy for type 1 diabetes? We're doing all those things because they're all cases where the human biology is absolutely clear and there's a new modality and no one else is doing it and the patients have great need. So these are the programs we have. Another difference in our approach is many companies will have what's called shots on goal. It means throwing things against a wall and hoping something sticks. The idea is that if you do more things, maybe more things will work. That assumes that you can do more, that like good ideas are abundant and the only problem is processing them. I think that our view is good ideas are really hard to find in terms of human biology, therapeutic potential, unmet need. And then when we pick a few of them, we actually put all our resources on them. So even though we are not a small company at this stage, we only have like 8 projects. And so as opposed to like some big pharmas I know have 400 projects in research, we have like 8. But those 8 get fully resourced to try and figure out how to do it. And it's as I described. So with that, I'm going to turn from sort of the framework to now just going through 3 different stories, just to give you a sense of what I'm talking about. The first is a story that does come from UCSD in some ways, because it's the story of cystic fibrosis and the work of our San Diego site, which is a mile or so from here, that was founded as Aurora Biosciences by Roger Chen, Charles Zucker, 1996 or so, and then Vertex acquired in 2001, 22 years ago. And although the drugs were discovered at Vertex, I thought, given where I am today, I would give the shout out to UCSD and Roger Chen, who I never had the pleasure of meeting, but Paul Negulescu, who was like employee number 4 at Aurora and led the site for 20 years, still speaks so warmly of Roger and those early days. So cystic fibrosis, you can tell I like history. Cystic fibrosis was discovered in 1938. It's not like it's been known forever. It wasn't handed down in the, in the Bible. It was actually a woman named Dorothy Anderson, who was one of the first female professors at American Medical School, a pediatrician and pathologist at Columbia, did a series of autopsies on children thought to have celiac disease and discovered cystic dilatation and fibrosis of the pancreas, the lungs, the liver, and said this is called cystic fibrosis. It was rapidly figured out it was an autosomal recessive disease. And for 50 years, its cause was unknown, but supportive care improved the lives of people in terms of like helping with first better nutrition and then better lung care and antibiotics and all like. But it was in 1989, the first autosomal recessive positionally cloned gene. So Duchenne muscular dystrophy and chronic granulomatous disease, 1986, 1989, the CFTR gene was cloned. And it was called initially the cystic fibrosis transmembrane regulator because no one knew what it did. But then within a few years, it was shown it is a chloride channel. And that really explained the disease because the disease was known over those 50 years to have the following properties. It caused thick, sticky mucus in the lungs. It caused that thick, sticky mucus also blocked pancreatic ducts. It caused malnutrition through poor absorption. And also children with this disease had salty tasting sweat. And it was figured out actually just before the gene was cloned that that was due to a chloride transport defect in sweat glands. So when it's figured out this was a salt transporter, it made perfect sense. Absence of this salt transport, dehydration of mucus, blockage of ducts in the lung, the pancreas, et cetera, leads to cystic fibrosis. And it is a systemic disease. And it's a very bad disease. At the time when the gene was cloned, the average life expectancy was probably in the 20s. And it had actually been like in the, it was like 10 years initially, and then it rose to the 20s. And I'll tell you later what it is today. But supportive care definitely added decades, a couple of decades to life, but it was still at this time a very serious life-threatening disease. I will also note, just for the geneticists in the audience, you see the child with cystic fibrosis on the cover of Science in 1989. What are the red, orange, yellow, green, and blue sort of abstract art that looks like whatever the art? I love the art on this campus, by the way, it's really fantastic, but sort of looks like an abstract artwork. Does anyone know what that is? What are those lines? They're haplotypes. They're the haplotype because the way that the CFTR gene was cloned was for position, there was linkage, but there was actually a haplotype-based association study. So like the HapMap project, which was often discussed in the heat of the genome project as somehow coming out of technology, actually came from the HLA and also cystic fibrosis, where Aravinda Chakravarty was sort of the person who recognized how to use haplotype mapping to find the CFTR gene. So my colleagues at Vertex 20 years ago, 1 mile or so from here, it led for the entire time by Paul Negulescu, who's sitting right up here on the right and is probably on site this morning, and Fred Van Goor, who's the head of biology. 20 years ago, he was hired. And this is Sabine Hadida, who's the head of chemistry. And they still all work together. And they discovered every CFTR modulator on the market or that's ever progressed past phase 2 in this field. And they worked on it for 22 years. In 1999, 24 years. In 1999, Paul Negulescu wrote a proposal to the Cystic Fibrosis Foundation saying how he thought you could use small molecules to treat CF. This was 10 years after the gene had been cloned, and every effort to that point had been based on genetic therapy. But then, as now, no one has ever succeeded in doing a genetic therapy of the lung. And he had the idea inspired by the understanding of the cell biology of the disease and also a critical experiment done by Mike Welch at University of Iowa, where he did a temperature shift experiment on Delta F508, the common variant that has that haplotype, where he showed that if you lowered the temperature of human bronchial epithelial cells from 37 to 30, the protein, which was sequestered inside the cell, would rise to the surface. And Paul had the insight, I think others thought of it as well, I'm not sure he was the only person who thought of it, that temperature shift, which is a classic model system geneticist experiment, told you there was some thermodynamic issue with the protein, not that it was completely gone and not that it couldn't be stabilized. And we thought maybe a small molecule could stabilize it. And so this grew out of an understanding, as I said, of the biology of cystic fibrosis, which was that the CF sort of goes from like understanding the disease, basic care, positional cloning, find the gene, figure out the function of the gene, and figure out the mutations in the gene. And the mutations it turns out there's many of them. There are a small number of people, less than 10%, who carry a mutation that gets to the cell surface. That's this pumpkin-looking thing at the cell surface is the CFTR protein. Protein gets to the cell surface, but doesn't open to transport chloride. Those are called gating mutations. There's less than 10% of people who have 2 copies that don't make any protein. So it's just like a null mutation. But the vast majority of people have 1 or 2 copies of a mutation called Delta F508 that has 2 problems. It turns out, first, it doesn't get out of the protein processing machinery. And then if you lower the temperature to 30 degrees, it'll get up to the surface where it has a gating mutation. So Paul's proposal in 1999 was we'll make 2 drugs. One will get the protein, stabilize it, get it to the surface. The other one will open the channel and make it transport chloride. And we can use the gating mutations to develop the first one and then the other one. That is exactly what happened. And all the drugs on the market today come from that proposal he wrote in 1999 and moving it forward. So I'm going to tell you first about what I like to think of as testing the CFTR modulator hypothesis. The CFTR modulator hypothesis was the idea that if you could make a drug that would restore function of CFTR, it would be useful to patients. This was not believed to be true in the field. I know that because I taught genetics at Harvard Medical School when this period of time was going on. It had nothing to do with Vertex. And every expert I knew said, genetics-inspired drug discovery makes no sense. It's all developmental. And anyway, even if you could do it, there's already scarring and infection and it's too late. And so not going to be useful. So the key thing was to actually make a medicine that could restore CFTR function and see what happened. And the idea here was the mutations that weren't open to find a drug that could. And there's a mutation called G551D, which is rare, but there's a few thousand people with it. So you could use that. It has a pure gating defect. So after 6 years of work, they had identified through screening using proprietary methods that came out of Aurora that had been Roger Chen's novel methods for fluorescent readouts in cell biology. They found medicines, found a drug now called ivacaftor that could restore function of CFTR in human primary bronchial epithelial cells to sort of 50% levels, which made sense because they had 1 copy of G551D. So you could sort of get that allele working better. And when you looked at these cells and using chloride transport as a readout, you could see about 50% function. If you ask the people who did this work, when were the exciting moments? One was when they looked under the microscope at cells with this agent. And if you look, you can see normally, you see how if you look on the left where it says no ivacaftor, it's just sort of a flat plane of cells. If you looked at normal cells, you'd see wiggling because there's actually cilial beating that moves the mucus along. When they added the ivacaftor, if you can see the wiggling there, basically immediately the cilial beat started. That told them the machinery was all still there, at least in these cultures. And the question is what happened in patients? And again, the expectation was that there'd be no effect. And one of the things that made this program seen as an untenable commercial idea was that there'd be no effect and it would just slow the rate of decline. But it turned out you had to do the experiment to find out. And within 2 weeks of starting ivacaftor in the people who had this gating mutation, there was a dramatic increase in lung function as measured by forced expiratory volume at 1 second called FEV1. FEV1 happens to be the regulatory endpoint. And to put this in context, people who have CF lose 2% of FEV1 every year. It's actually normalized to your age and gender. So if we have no lung disease, have 100%, and these people lose 2% per year. So by the time they're like 20, they might be at 60% or 70%. So seeing 10% come back means like that what you've lost over a lifetime, these were adults, like a third of it comes back within 2 weeks. And then it's stable, it doesn't change. I'm now going to switch though. That was only a few thousand people. That drug was approved for the first time in 2012. It's now been on the market 10 years. And for time, I'm just going to talk about the rest of the drugs and I'll give you the long-term outcomes at the end. The key question was how could you get to all patients with CF, or at least the 90% who made a protein? And that was a harder problem because like there'd never been a medicine made that could make a protein, refold. And they did it by screening in the presence of the potentiator. So they could then have opened the channel and they screened for things that could restore function at the top of the cell surface. This was a heroic effort. It turned out to require not 1, but 2 different correctors, they're so-called. And just to give you a sense, a typical drug discovery program in big pharma might have hundreds of molecules synthesized, millions screened, then you get what are called hits. And then a molecule made is like a chemist. They make a hypothesis, they make the molecule, they test the molecule. This program overall is now up to 40,000 molecules made. And the second generation corrector, which was discovered in 2016, required tens of thousands of molecules because it was just such a hard problem of drug discovery. And I just want to mention, because a lot of people think drug discovery is all about potency and maybe AlphaFold's going to help. It really doesn't understand the problem. The problem is not how do you find a molecule that binds to your target. The problem is how do you find a drug? A drug is something that is orally bioavailable, if it's a pill, safe, can be manufactured, doesn't have drug-drug interactions, has stable metabolism. It has to do nothing else and do the thing you want. It's called multi-parameter optimization. It took tens of thousands of molecules to find the ones that today exist. When they finally found the multiple combinations, I'm sort of summarizing 20 years of work in a few slides. The data on the left is from a 2018 New England Journal paper, where you can see that using that same assay, a human primary bronchial epithelial cell from patients, but in this case, having one copy of Delta F508 and the other a null allele. So that represents 90% of people have either one or two copies. When you added ivacaftor alone or tezacaftor, which is the first generation corrector, you could get some function. When you add all three, you could get higher levels of function than we're seeing with ivacaftor in G551B. And then in the clinical trials, what's been seen is about a 14% improvement in acute lung function, which is again on average about half of what people would have lost over their lifetime. And also the measure of sweat chloride, which is a direct measure of chloride transport moving below the diagnostic threshold. This medicine is now approved in 40 countries around the world and for genotypes that encompass 90% of CF. And recently, Jude Chen at Rockefeller has published cryo-EM structures of our molecules on the protein. And there's a lot of unpublished data done at Vertex, but we didn't publish it. So Jude Chen gets all the credit. But the bottom line, but then at least I can show it to you because Jude Chen published it. So if you look here, what you can see is the CFTR protein, and you can see the three molecules, ivacaftor, tezacaftor, and alexicaftor, and they all bind at three different sites on the same protein in the membrane, unprecedented, and explains why the molecules tend to be lipophilic, which was very difficult. And no other company, people used to laugh at Vertex molecules because you're supposed to make drugs that are highly soluble. And these scientists in San Diego were like, I can't make a drug that's soluble. So can you figure out a way to formulate a drug that's not as soluble? When you get the cryo-EM structure, you understand why they're lipophilic, because they're binding in the membrane. The sites that exist are in the membrane. And they bind very far away from the mutation. You can see Delta F508, and you see they're all up in a different place. So how do they work? Well, one of the things about how they work is basically the way they work is they stabilize CFTR and treat many, and they not only do that, but they don't just treat one mutation. It turns out there are hundreds of rare mutations, and a large fraction, but not all of them, respond to these medicines. And that was a real puzzle. Like, why do they? It didn't have to be that way. If you think about quote unquote personalized medicine, people might think you're treating the mutation, but it turns out it's not what's going on. These molecules, and I think, the way it works actually is like tezacaftor binds to an intra-domain binding site on the outside of this protein. And alexicaftor works by binding multiple different domains and basically just changing the thermodynamics of their interaction allows the protein to get through protein processing and get to the surface where the potentiator opens them. It's totally remarkable. So what is the impact of these medicines on patients with CF? We now have, as I said, 90% of people of the age groups yet studied. You have to start with adults and work your way down. But of people who are of the ages studied, which is now 6 and above, we have 6 and above for the triple therapy, and 1 and above for double therapy, and 1 month and above for Kalydeco, because they've been on the market longer. We've made it down to lower ages. It's now 90%, and was said we're approved and reimbursed in 40 countries around the world. There's real world evidence and follow-up, and the most recent data is from 16,000 people in the US. This is by the Cystic Fibrosis Foundation, not by Vertex. And in that data, the mortality reduction with Trikafta is 75%. The reduction in transplantation is 87%. The reduction in hospitalization is 80%. And a recent study that was published looked at the projected survival. It's a model based on the changes that are being seen. And just to give you a sense, in the absence of these medicines, the median age of death is today thought to be 38. In the presence of these medicines, it's predicted to be 71. And in that same paper, they projected if you start at 12 and above, and remember we're approved now to 6 and above, it's 83 years. So these actually are having a profound effect on the lives of people with this disease. So I do want to make clear that this required a very large effort. It required 20 years of work at Vertex, but also required a tremendous amount of work with the CF community. We put 13 compounds in the clinic. We've done 180 clinical trials, enrolling in total, not necessarily all unique individuals, 11,000 people. We have 4 approved medicines, and we have another in phase 3, which is the next generation beyond Trikafta. And so it's not a small thing, but it has actually, I think, resulted in a significant impact. So I have a few minutes. Oh, I guess if that's right, I have a little bit of time left. So I'm going to talk about 2 other diseases. That's CF, and it's a very exciting program. I want to talk about two other diseases that we're making progress on. One is sickle cell and beta thalassemia. Sickle cell disease was was known for a long time, but the mechanism is famously sort of described in this paper by Linus Pauling called Sickle Cell, the First Molecular Disease in 1948. If any of the students or fellows haven't read it, I recommend it. It's about where they took a protein and they ran it on a gel and they saw a different migration on a gel. And this is the first time, to my knowledge, that a gene was related to a protein was related to a phenotype, like in the world. So that's pretty cool. And then in 1956, Vern Ingram discovered that it was a single amino acid change and caused sickle cell. And we've known that now for 70 years, but we didn't really have an effective treatment. And in a way you could say what was obvious what you'd want to do, you might want to change the DNA and get rid of the sickle cell mutation, or you might want to do something to the binding of oxygen. But another, and the disease itself, I should say, and there's another disease called beta thalassemia, which instead of that one mutation is a null essentially, or knockdown of the hemoglobin protein. Stu Orkin, the same Stu Orkin I mentioned earlier, and will mention in a minute, figured out a lot of the mutations in beta thalassemia in the 1980s. So these are very significant diseases. The prevalence of sickle cell disease is in the US much higher, is higher than CF. There's about 100,000 people with sickle cell in the US, and much higher around the world, with millions of people around the world. It has a very high rate of morbidity and mortality. The age of death is about the same in sickle cell and CF, so there's similar severity. And in sickle cell in particular, it's just ongoing pain crises, very severe, very difficult for patients. One clue that turned out to be the one we followed has to do with hemoglobin F. So there's something called fetal hemoglobin that developmentally is on in fetal life, and it's there because the fetus has to get oxygen from the mother and has higher affinity for oxygen, and it turns off in the first 9 months of life. And it was known that fetal globin could substitute for sickle cell or thalassemia because children who are born with it don't get sick until the fetal globin goes away. And there are some people who have hereditary persistence of fetal globin who have higher levels of fetal globin, and they don't get sick, even if they have a sickle cell or thalassemia mutation. And then there was a genome-wide association study done by Stu Orkin, another one done by Gonzalo Abacasis and others, that identified a number of variants, common variants in the genome that could affect fetal globin and also severity of sickle cell and beta thal. So one of the things about this kind of target identification is it was known from the first paper or the first year that not only did these variants affect fetal globin, but they looked in people with severe sickle cell and natural history studies and showed they had positive outcomes with, compared to people who didn't. So Stu Orkin's lab figured out what BCL11A does and how the variant works. And in particular, what he discovered is there's an erythroid-specific enhancer in BCL11A that regulates BCL11A. BCL11A is a repressor of fetal globin. And if you have a change in this erythroid-specific enhancer, then you end up with more fetal globin. We decided to work on this in 2015. We partnered with CRISPR Therapeutics. In 2021, we published the first 2 people ever treated with CRISPR for a human genetic disease for sickle cell and beta thalassemia. And the strategy was basically to mimic human genetics. We took the finding from genome-wide association studies about BCL11A, and we used CRISPR to make in an ex vivo gene editing experiment the same change or, at that location. We also did a lot of work. We screened through thousands of guides to find one that didn't have any off-target effect, which we published in the journal, and also to work on the conditions for growing the cells and doing the manufacturing to make sure we got the long-term repopulating stem cell. Because it turns out if you don't get that, then you can show in a dish it seems to be working well, but you won't actually have a long-term treatment for patients. And there's no marker for the long-term repopulating stem cell, and it's a tiny minority of your drug product. So it takes a lot of work to try and figure out how to do that. We announced last year just about a year ago at the European Hematology Association, we'll actually be giving an update later this year on the current status, a total of 77 patients treated with what's now known as exa-cel. 42 of 44 patients with transfusion-dependent thalassemia were transfusion-free in the period of time of follow-up, and the other 2 had dramatically reduced transfusions. Of the 33 patients treated with sickle cell disease, none had a vaso-occlusive crisis. The average was 4 vaso-occlusive crises per year on entry. And the safety and tolerability were comparable to that seen with the busulfan conditioning regimen used in this bone marrow transplant. So that's very promising, and we reported last week that we had completed filing for approval in the US, in England, and in Europe for this therapy, which has the potential, if it is granted, if it is approved, to be the first CRISPR-based therapy. And then the last story I'm going to tell is about another finding from genome-wide association studies called APOL1-mediated kidney disease. which is a genetic kidney disease. This was discovered, actually, the first localization of this chromosomal region was actually from an admixture mapping study, which is a form of sort of linkage analysis. And but it was a very large region. And actually, originally the gene was misidentified as to what it was. But then Martin Pollock in this 2010 science paper showed that variants in APOL1 had a dramatic increase in risk of kidney disease. And just to give you a sense, APOL1 homozygosity, it's only people who are homozygous, who have 2 copies of these variants. These variants are very common in people of African ancestry. And to give you a sense, about 10% of people carry 2 copies of these alleles among populations with West African ancestry. And in people with focal segmental glomerulosclerosis, it's up to 75%. In people with HIV-associated nephropathy, it's a very high proportion. In people with what's called hypertension kidney disease, which is really just kidney disease of no known cause, it's a 2 to 3-fold increased risk, and in lupus nephritis. So this was found to be a significant multifold risk factor for kidney disease, but what this protein did was not known. The only clue was, one, it's a component of cholesterol particles, which we don't believe it is relevant, and it's a protective factor, these variants, for trypanosomal sleeping sickness. And actually, that had been worked out before. The APOL1 variants that turned out to be associated with kidney disease were known to be protective for this form of trypanosomal sleeping sickness. So, and they're also very uncommon outside of Africa. And it's a little bit like the sickle cell story. It's thought with malaria, that where the trypanosomal illness was prevalent, these mutations, which protect from certain strains of trypanosomes, had become more common. So in 2015, when I moved to Vertex, we decided to start a project on this. We collaborated with Martin, who I had known for 25 years. I used to walk home from lab with him when I was a graduate student. He was a postdoc. And we decided to collaborate and worked with him and his colleague, David Friedman, who's great. And we really, no one knew what the protein did, but we decided to follow the genetics. So the genetics told us the natural selection that the function was having to do with a poor function, having to do with protection from trypanosomes, and we decided to follow that. So I would say before we get there, the natural history of people with 2 APOL1 alleles is they progress to end-stage renal disease about twice as fast as people with the same clinical presentation who don't have 2 APOL1 alleles. So it's not only a increased risk, but it's also a more severe form of the disease. And just to give you a sense, if you look at the x-axis on these plots, what you can see is that over a period of about up to 50%, about twice the rate of progression, and that like a high percentage of people will end up with end-stage renal disease in the first 5 to 10 years after diagnosis. So we came up with this hypothesis, which was this had to do with the poor function that was already implicated in the, and we hypothesized it was known that the protein was expressed in podocytes in the kidney, and those were the cells that were damaged in APOL1 kidney disease, that it was poor function in the podocytes. So we developed 6 different assays in-house that measured everything from binding to the protein, to inhibiting its poor function, to protecting from trypanosomal illness, to making a mouse model that expressed APOL1 and could get the disease. And we published in the New England Journal about 2 weeks ago, the first phase 2 study, the preclinical data, and the first phase 2 study for the first APOL1 inhibitor. And remember, there were lots of people who said, what is it? Why would you want to inhibit this thing? Like, they already have kidney disease. It's too late, or there'd be no effect. And honestly, we didn't know either, right? Because no one had ever done it before. We'd done it in a mouse model, but not necessarily telling you what's going to happen in people. So you can look at the preclinical data. It shows it is a selective, specific binding to the APOL1 channel and blocks its poor function. And then we showed in 16 people with severe kidney disease who had high-grade proteinuria, 2 APOL1 alleles, still had kidney function, and no other obvious cause. And they all had, on histology, what's called focal segmental glomerulosclerosis, which is a severe presentation, that there was a 47.6% reduction in proteinuria in the first 13 weeks, which is the largest reduction in proteinuria ever seen in a focal segmental glomerulosclerosis study. This is only in people with 2 copies. And as you can see from the waterfall plot, as it's called, all but 1 patient responded out of the 16 patients. This study is now in pivotal studies, phase 2, 3, and we're enrolling and hoping to see if we can replicate this. So in short, I told you that our goal was to actually sort of break free of the model that says we have to pick a tissue or pick a platform. And we picked a set of 6 programs that have made it beyond CF into pivotal trials. Yeah, I'm saying to phase 2 trials, sickle cell beta thal with CRISPR, a pain program with selective sodium channel inhibitors that was started at Aurora before CF was, focal segmental glomerular sclerosis, APOL1, the type 1 diabetes cell therapy, and an AAT program. 5 of the 6 had positive proof of concept, are now in pivotal development. The 6th one had positive proof of mechanism, but actually is too small an effect, and we're going back with more potent and effective medicines. So I've been arguing for like 15 years as an academic and then at Vertex that it is actually possible to succeed at a higher rate. You just have to pick the programs in a different way. But honestly, I was a pundit, right? And a lot of people didn't agree. And there's lots of other ways to do drug discovery. And a lot of investors, because when you're at a publicly traded company, investors comment really like, why would Vertex succeed at any higher rate? But I think we now have some empirical data to say that we can actually succeed at a higher rate if we don't start with my project. Note that no genetics I ever did contributed to these. I would start with my project or my pet hypothesis or my thing or my platform and just survey the world and say, what is the best project you can find? And then put a lot of resources on it. You might be able to make progress. And the last thing I'll stop with, and I know I'm out of time, is I call the talk Humanizing Drug Discovery for 3 reasons. The first is sort of obvious, my belief that human biology is key to medicine. And as silly as that sounds, let's all be honest, there's a tremendous amount that goes on that is not based on human biology. And it's very hard to do human biology, but I believe that it's worth it for patients. So I think that's important. The second thing I wanted to say, I call Humanizing Drug Discovery, is because the people who do the work. One thing about scientists in industry is they don't seek fame, they don't seek attention, they don't publish their papers always, and they don't get known. I have never met scientists better than, I'm not saying that the people at Vertex are better than the people at UCSD or Harvard or MIT, but they're the same. They're at the same level of excellence. They just don't get recognized because no one knows who they are. So I try and tell you a little bit about who they are. But the most important aspect of humanizing drug discovery goes back to Francis Weld Peabody, focus on the patient. And the picture here, which is of Victoria. Victoria is the first person treated with a CRISPR-based gene editing therapy for sickle cell disease. She chose to talk to the media, not through Vertex, but she talked to NPR. There are 3 NPR stories about her. She's a remarkable person, just watching her. And there's this video of her before, which she's sitting on the bed waiting to get the infusion of the CRISPR cells. And she knows no one's ever done this before and no one's ever been treated with this disease before. And she says, I don't expect this to work for me, but I hope it creates hope for others. And I was so moved by that, the idea of someone so courageous that they would do that, not thinking it would help them. And it's very inspiring. I'm glad to see that Victoria seems to be doing well based on her interactions with the media. And it does inspire us every day. And thank you for the time and the chance to share the work with you. Yeah, if there's time, I'm happy to take questions. In the room, yes, I'll start here and then we can go online. Yes.
I was just wondering about the 25,000 molecules you made. Would that have been less if you had known the crystal structure and where it was binding ahead of time? Were you trying to optimize solubility?
I think yes, I think that had we had all the information we have, we could have done it more. It wouldn't have been 10 molecules or 100, but we could have done it better. And we ourselves solved crystal structures and did mutagenesis and SAR to make the next generation of molecules. But what we've learned is that the tools are not yet there at the level of precision you need for a previously undrugged protein. So like having done all we did, we can now learn and do better. And in fact, we've now incorporated cryo-EM and AI and all these things into all our drug discovery programs. But we find that if you just start, like you might read in the paper, AlphaFold's going to do everything. It's just not true. But once you actually have a cryo-EM structure and liganded structure and you start using all these tools, you can definitely accelerate progress. But the thing that no one has yet solved, and I think will eventually be solved, but it's far away, is safety, PK, bioavailability, drug-drug interaction. So it's actually kind of silly, if you want to know the truth. All this talk about AI drug discovery, it's as if finding a drug was finding a binder, but anyone who does drug discovery knows that's like the first step in a marathon. And so I think there's just a misconception of what a drug is. They think it's like a binder, but it's not. A drug is something that's safe and effective and stable and bioavailable and manufacturable and all that stuff.
I wanted to follow up with one other question, which is the sickle cell, obviously really, really exciting, but what about all of these, I'm sure you get asked this, all these people in Africa that make $1 a day? And And are you going to continue to look for cheaper therapies like small molecules that might help
Yes,
people who can't afford stem cell
we
transplant?
are deeply engaged in that. So if you ask me what I think is the goal, it's to make a therapy that's highly effective or multiple therapies that can treat as many people as possible, including around the world. I think what we've really done with XSL, and there was a paper in New England Journal from David Williams' lab at Children's that did it with an RNAi, is we have validated the hypothesis from human genetics that if you modulate BCL11A, you can create an effective therapy. So we are working both on improved ways to deliver what's called XSL now, for example, improved conditioning or in vivo editing or something that could make it a less invasive procedure. But we've also had a large team working for 5 years on trying to make a small molecule to do the same thing. And we haven't done it yet. We haven't succeeded yet, but I'm a big believer that if you have really smart people and a really important problem, you can eventually get there. And that's actually, I think, the more likely path to get to everyone around the world is to find a small molecule. And I was talking with Joe earlier about how in spinal muscular atrophy, there's a oligonucleotide therapy, a gene therapy, and a small molecule therapy. Once you crack a problem and you have a target, there's multiple modalities that can get there. So I'm hoping, sort of one of the things that keeps me going is like, I want to find a small molecule so we could do that, but we haven't found it yet.
David, that was great. I've had friends in industry tell me that there are a lot of incentives aligned to do Me Too drugs rather than blaze a new trail. So FDA approvals, reimbursement things, all that. How do you guys sort of always swing for the fences and seem to blaze new trails like that? How do we encourage other companies to do that?
So it's a strategy, like strategy is sort of like the battles you choose to fight. And Jeff Leiden, who is the chair of Vertex now, was the CEO when he hired me in 2015 and recruited me to his board in 2012. This was his vision. Like he had been a leader of Abbott. He had been a venture capitalist. He was a very successful academic for many years. And he had the vision of a company that wouldn't do the Me Too thing, wouldn't just try and minimize risk, but would also be very science driven. And that's what attracted me to Vertex, was that vision. So I think part of it is it's actually our strategy. I think that the reason I put that slide in, which for people who don't follow the industry as much may seem funny, is those are the three strategies that really exist in the industry. There's like, one is a platform strategy, tends to be an early company strategy. One is a commercial strategy based on where you have salespeople and marketing and distribution. And the third one is just copy people, let them blaze the trail. Certainly not, we're not going to, it's not our strategy. But I think that, I think there's tremendous value that can come to everyone, to society, to patients, and to stockholders if you succeed at it. But you have to be really good at science and really committed. And most people don't believe it can be done. And that's why I'm not trying to like pat us on the back when I say it can be done, but I am trying to convey that, like, because there's a broad-based nihilism, which is that nobody can predict what's going to work and it's all just failure, and occasionally you succeed and then you jump on it. I object to that concept.
Just the ApoA1 is really interesting. So does that drug inhibit the transport of cholesterol, or does it prompt the destruction of that specific mutated copy, or the two mutated?
No, it's a good question. I mentioned that it is carried in the blood and cholesterol particles. There's no change in the clinical trial on anything cholesterol related. We think that's just irrelevant as far as we know. The drug sits in the membrane of podocytes, and it transports an ion. It's not 100% certain what the ion is. We use thallium and things like that to mimic it. And our drug is a highly potent and selective blocker of the pore. So again, thinking about target validation, what we actually do is test the pore hypothesis for ApoL1. There are other hypotheses. If you look, you can see other companies were thinking about, like, antibodies in the blood or whatever. But I think what we showed was it's the pore function, at least, is adequate to make a benefit.
In the chat, Paul Insel asks, says, great talk. Cindy Brenner once said the next model system is human. Do you think that every drug discovery program must be based on human genetics?
Okay. First of all, I do not think that every drug discovery program needs to be based on human genetics. And in fact, like, our type 1 program is not based on human genetics. I do believe that every program that has a reasonable likelihood of success should have strong basis in human biology. And by human biology, I mean like genetics, pharmacology, transplant, there's a lot, endocrinology, there's lots of ways of doing things. I don't believe correlation is that helpful. So like, so many things change in the body that saying like this is expressed higher or lower in this cell is just not that helpful. And I want to be clear, I'm a big believer in model systems. I just think that for, first of all, to learn biology, to learn how things work, and to generate or test hypotheses. I just think if you're going to embark on a decade-long effort to make a drug and put people on a medicine to test it, you should at least think about the human biology. And it's just not the way people are trained. You know what I'm saying? Like, I will tell you, I mean, I was a professor for 20 years before I went into industry, and I said all the same things as Amit knows because he was in my lab, but it was like not, people didn't agree. People were often quite annoyed at the idea that you would sort of say that for all they said, well, doctors say that, but doctors are trying to take away the money from basic science. It's really just to the extent that society invests in us because they believe we're going to do something relevant to medicine. We should just always ask ourselves the question, is it really relevant to medicine, or is it just what I'm interested in? And it's okay if it's what you're interested in, but it's not necessarily a good basis for making a medicine or exposing patients to it. And I actually think if more people tried to work on human biology, we'd know more human biology. You know, like what troubles me is it's sort of, I often get the question from people, how do I get the human biology to prove my target's a good one? I'm like, I can't make human genetics prove your target's a good one. When I used to consult for pharma, that's what they wanted me to do. Like, can you get a human genetic basis for my target? Like, I'm sorry, I can't do that. I'm not like evolution. You know, I can't do that for you. But you have to really ask yourself the question, are you trying to start with the goal, which is make a medicine for patients or diagnostic? And are you following the science? Or are you starting with something else and then trying to prove that what you did is helpful to patients? They're just very different things.
Any other questions? Thank you, David.
Thank you for having me. It's a great pleasure.